Okay, so as Dan just nicely illustrated, coral reefs are under threat from a lot of different sources, but principal amongst those is rising ocean temperatures that cause corals to bleach. And they bleach when they lose their algal symbionts that provide a lot of nutrition for them, and if they don't get those back fairly quickly, then they die. And so that's probably not surprising for most of you here that corals are under threat. To me, the extent to which they're under threat is really surprising. So in the 2016-2017 bleaching event, something like half of the corals on the Great Barrier Reef died. Not bleached, but died from that bleaching. And some of those were replaced by recruitment and growth, but like not that many. And those reefs are super diverse. They support a huge number of other organisms. And so recent estimates say that we're likely to lose 90% of reef corals by 2050, which is like real soon, like within our lifetimes. And so that sort of makes me feel like this. And so we know that that's a possibility, but there's lots of ways that corals could respond to climate change. So the worst of those is extinction, but they could shift their ranges. That might happen, but it takes hundreds of years for that reef structure to build up. So in the short term, that's not a great solution. They could acclimate through behavioral or physiologically plastic mechanisms. That's a possibility, but they're kind of already at the edge of their thermal temperature range, and so the extent to which they can do that is probably limited. And so we're looking at whether they might be able to evolve, and can they adapt to these changing ocean temperatures. And there's some potential that this might occur. So if you look often at a bleached reef, you'll see that many of the corals are bleaching, but not necessarily all of them. And so you can see some of these corals are white, some are still brown, indicating they still have their symbionts in them. And so what's happening there? Like they're all in about the same environment. They're all experiencing the same temperature there. But maybe those coral individuals have different genes. Maybe the symbionts inside of them have different temperatures, different temperature tolerances. And so there's some potential that there's genetic variation there. And so because there's a genetic variation, there's a potential for evolutionary rescue. And evolutionary rescue is this idea that red line is the abundance of a population. And as that population starts to decline, they're experiencing more and more selection pressure. And that blue line is the frequency of adaptive alleles in that population. And so over time, as the population declines in abundance, the frequency of adaptive alleles goes up, and the population might recover in abundance due to those adaptive alleles. And that's a potential that this could occur. There's a big push to say, let's not wait for this to occur, and let's do assisted evolution on reefs. And so let's go into the lab and breed super corals or super symbionts and seed reefs with those and sort of save them that way. And there's an ethical debate about that, and the proponents of this would say, yeah, there's a risk to doing that, but we're in a desperate situation, so it's worth the risk. So we're interested in just whether there's the potential for that to even do this assisted evolution process at all. And in order for that to happen, you have to start with some genetic variation. There has to be those adaptive alleles in the population already. So that genetic variation has to already exist. But then we also have to think about that as things evolve, they're not evolving in a single environment where they're the only species there. They're evolving in response to many other species too. So this is the idea of Darwin's tangled bank, that there's many species interacting, and they're all influencing how each other evolve. And so we're interested in that as well. So we're using, sort of as a model for stony corals, we're using a soft coral, Untitled Chapter. It houses mostly one symbiont of the Symbiontaceae family. And so we're looking at interactions between these two. And what's nice about the system is we can grow these symbionts in culture in the lab, and the stuff that we're growing in culture is representative of what's in the host in the field. And so we're just looking at these two species. We're also thinking about other species as well. So Chelsea Brisson's looking at the bacteria associated with this interaction. So she's giving a talk today at 4:45 if you want to see what's happening with the bacteria that turn out to be hugely important as well, unsurprisingly. And so in terms of this evolutionary rescue, we're thinking about not just can corals be evolutionary rescued, but can the symbionts evolving, can we save the corals that way? So can symbiont evolution cause the evolutionary rescue to occur? And in order to do that sort of assisted evolutionary rescue, we need several things to happen. So one, we have to have that genetic variation there in the first place. We'd like to be able to do laboratory selection experiments where we can select on the traits that we want, specifically thermal tolerance. And then those selected traits, those things we select for in the lab, they have to affect the host as well. So, and that's a key component of what we're doing. And then additionally, if you want to outplant these into the field, then that whole holobiont has to be able to reproduce itself. We don't want it to be continually rescuing the reefs. We'd like them to become self-sustaining. And so I'm not going to talk about that last part today. Jenica at my lab had a poster on this on Saturday, and so if you're interested in that, she's introducing these symbionts back into hosts and looking at reproduction of those holobionts. So you can chat with her about that. So first question is there genetic variation in response to temperature? We just took a bunch of different genotypes that we had in culture in the lab, put 10 genotypes there, but it's actually only 5. And we grew them at 26 degrees, which is kind of ambient temperature on a reef, and we grew them at 30 degrees, which is a stressful temperature for a lot of corals. It's often the bleaching threshold for many corals. And then we measured growth rate, photosynthetic rate, and respiration. 3 traits that we think are likely to affect interactions between hosts and symbionts. Growth rate, probably good at some point. It's hard to say whether it's always going to be good. If you're the host, you might not want your symbiont always growing as fast as possible. But photosynthetic rate should be, that's where they're generating the carbon to provide to the host. And respiration rate is probably, a high respiration rate is probably a detriment to the host because it's using a lot of that oxygen. And so this is not real data. This is just what we were hoping might occur, is that we would see different genotypes, or the different colors there, and that they might respond differently to temperature. So the blue genotype is really strongly affected by temperature, but the green genotype is affected much less. And if we did this a different way, doing statistical analysis, we could look at the variance components, and if we see a temperature by genotype interaction, that sort of G by E interaction is what tells us there's potential for selection to occur there. And so we're hoping for large G by E interactions. And if we look at something like growth rate, we find that different genotypes respond differently. They all respond negatively to increased temperature, but some less negatively than others. And so you might expect those blue genotypes to be selected for at ambient temperature, but something like that orange genotype to be selected for at a high temperature. And if we look at the variance components again, there we find an 18% of the variation explained by that G by E interaction. So not a huge amount of variation, but like something. So, and if we look at our other traits, photosynthesis and respiration, we actually find much bigger G by E interactions there. So 34% and 61% respectively. And that suggests that there's potential for selection in response to temperature on those traits. And so, and these are traits that we expect to influence the strength of that mutualism. And so at this point, we're pretty excited about this. Like we're, do these selection experiments in the lab, let's see the reefs with them, reefs are saved, we can all go home. And so we left that part of the experiment with this real optimism, but I have just something like 7 minutes left here, so obviously there's more to come. So we thought our next step was that we would do these laboratory selection experiments. And so we went back to the host, we just isolated symbionts out of the host, and we isolated them at 26 degrees and we isolated them at 30 degrees. And what we were thinking is that we know that that genetic variation exists out there, but it's not, they're not that temperature tolerant. So could we select for these at higher temperatures? The somatic mutation rate is pretty high in the species. And so we thought, well, maybe over time, we'll get lots of like mutations that might be selected for, and we would get these like super heat tolerant algae. So we let them evolve in those different temperatures for 2 years. Generation time is about a day or so. So we're letting them evolve for 500 generations or so, probably a little more than that. And then at the end, we take those ones that evolved in different environments and we grow them again at 26 and 30 to look to see whether they're locally adapted to those temperatures or not. And what we find is really disappointing. So here's the growth temperature. The ones that grew at 30 grew slower, just as they had before, but we see no difference based on what temperature they historically evolved at. And so we don't see that the ones that evolved at 30 are growing any better than the ones that evolved at 26. So that's a little frustrating. If we look at photosynthesis and respiration, we kind of maybe see some signs of local adaptation in photosynthesis. On average, they're growing faster, but the variance there is so high, we don't pick up that difference statistically. It's also weird that there's so much variance there, because if there was this really strong selection, you'd expect the lowest variance. where you saw the most selection. And so, but also that high photosynthetic rate is associated with a high respiration rate. So it's not clear that that high photosynthetic rate is actually beneficial to them because they're respiring really quickly as well. And so, you know, we did this selection experiment, like, didn't come up with like this really promising result. But at least, you know, from that first experiment, we know that variation exists. So maybe we can take that variation that already exists and try putting those symbionts back into hosts to see what happens. And so we did sort of the same experiment we did in the first one. We took all of our different genotypes. Here, the genotype number's wrong. I think there's 6 genotypes here. But we put them into polyps. And so actually we put them into the larvae of the host, because the larvae don't have any symbionts in them, so we can introduce the symbionts as we'd like. And then we measured respiration and photosynthetic rate as they're growing at these 2 different temperatures. And what we find is if we look at photosynthetic rate, we find that, again, temperature matters, but the big differences in genotype that we saw before don't exist anymore. So at 26 degrees, all the genotypes are photosynthesized at about the same rate. They're respiring at about the same rate, and the same thing at elevated temperature. So we don't see those strong G by E interactions that we saw before. They're now essentially nothing. They're not there. And probably it's because the hosts are doing something to control the performance of the symbionts, which is not terribly surprising, but we sort of didn't think about that doing this. And so it suggests that that variation that we see in nature, at least in culture in the lab, we don't see inside of hosts. And so it's unlikely to be beneficial in those. So we find that those selective traits don't affect the holobiont very much at all. And so what we think is that there's, you know, just looking at the lab specimens, we see lots of genetic variation that we expect to undergo selection and maybe get some evolutionary rescue there. But once you put these things inside of hosts, that symbiosis mitigates any selection that might happen there. And it doesn't allow that variation to be expressed. And so, you know, there's potential to do these sort of evolutionary rescue experiments. It's just that I think if you can't solve climate change, any of these big conservation efforts that we try to do are kind of meaningless because that's the big root of the problem. And so if we can't solve that, so like my favorite example of this recently is that Australia has dumped tens of millions of dollars into reef conservation efforts, and including assisted evolution. At the same time, in the back end, they've also taken tens of millions of dollars and put it into harvesting coal reserves that have never been harvested there before. So any benefit they're going to accomplish is offset by this like increase in climate change in response to that. So this is my frustrating message, and take a few questions.
By focusing on the G by E, it sounds like you're assuming that they need to evolve plasticity, but couldn't they just evolve greater tolerance at the high temperature? So just look at the high temperature condition and define genetic variation for these traits.
Yeah, well, I think that's what the G by E is, that we do find genetic variation at, well, okay, I see what you're saying, yeah, just look at the high temperature, do you see variation there as well?
Exactly.
Yeah, and we do in culture, but again, not in the host here, so, good point. Yeah.
Do you measure ROS production or anything like that?
No. No, and that would be interesting too, to see like, you know, if they are stressed out, why are they stressed out? Or if they're not performing well, what's the real reason for that? Yeah.
So historically, elevated CO2 levels and elevated temperature in the marine environment, large-scale extinctions of coral reefs always follow elevated CO2 levels. So what is the ultimate goal of the evolutionary rescue if we can't immediately stop these elevated CO2 levels? Or we're going to have massive scale extinction of coral reefs regardless. So what's the hope with the evolutionary rescue that we have? Hopefully that we have mitigated levels of CO2 and that we can sort of retract these, or that we're just going to save small little spots.
I mean, I don't have a lot of hope for it, I guess. I think it's like, maybe you could get, if you stop CO2 levels where they are right now, you might be able to get things to adapt to those current conditions. But then it's going to change again, and so it's just a red-green thing where you're constantly struggling to keep up. And at some point, we've exhausted all the variation that exists to adapt to those higher temperatures and CO2 levels. So, yeah. All right, enjoy your coffee.